Today we'll be talking about the synapsis of chromosomes in meiosis. So we've got individual 1 with these chromosomes, and you can see they look different. The black part that's underlined, those are the parts that look different. These underlined areas are the same. And we can see that this part includes the centromere, and these are switched. They're flip-flopped. And this is pericentric inversion. The goal of meiosis is to maximize pairing between homologs. So in order to do this with an inversion, we're going to be seeing an inversion loop, which allows all of these to line up with each other. If we want to know what kind of gametes we're able to make with these 2 chromosomes and this inversion loop, with a crossover occurring between the J and the K region seen with this blue X right here, we're going to want to follow each line and include the crossover right there. So to follow the first line, we're going to get MIJ with our centromere, KLHG. And that one is going to look just like the parent chromosome. If we follow the second line, we're going to get MIJ with a crossover here, which leads to K, L, and back to M, which does not look like the parent. And it's also got a duplication and deletions of the H and the G. If we follow the third line, we're going to get MLK with the centromere, JIHG. And that's going to look just like the other chromosome, the other parent chromosome. And then lastly, we're going to start from this side, and we're going to see GHLK, and then the crossover here, which goes to JIHG. And that's what it's going to look like. We're going to see a duplication here and a deletion of the M locus. These 2 red gametes are going to be inviable because of those deletions and duplications. When looking at our second individual, we see they have chromosomes that look like this, and they're exactly the same, so we are not seeing any abnormalities. When we try to look at what the crossover event between the J and K regions on this event in meiosis, we are going to see the crossover event occurring right here with chromosome number 1, chromosome number 2, and they all line up very nicely, so it's going to be a straight line. And if we look at the gametes that it's going to produce, they are all going to look exactly like the original chromosomes, because we can see how this goes here, and then these inner lines, they cross over at the perfect areas to give us these all viable gametes, because there's no deletion or duplication occurring. So this question here says, is there any expected difference in fitness between the 2 individuals? And the answer is yes. Duplications and deletions result when crossing over occurs in inversion loops, and these recombinant products are often inviable, meaning individual 1 has less genetic fitness than individual 2.